Hello students. In the last lecture, we saw biochemistry of blood group substances and inheritance of ABH antigens. Now we'll move ahead for medico-legal applications of blood groups. What is medico-legal? Something that involves both medical and legal aspects. Fine. As we have studied blood group, their biochemistry and inheritance, we also now understand that they play a very major role in human health. So, blood groups are involved in the following applications. First is blood transfusion, hemolytic disease of newborn, detection of Rh antibodies, identification of Rh incompatibility, prevention of isoimmunization, ABO hemolytic disease, blood group and diseases, and forensic aspects. Fine. Now let's begin. Blood transfusion. It's a routine medical procedure in which donated blood is provided to you through a narrow tube placed within a vein in your arm. This potentially life-saving procedure can help replace blood loss due to surgery or injury. Fine. The existence of several different blood group antigens makes it almost impossible to obtain perfectly matched blood for transfusion. So, in routine practice, ABO and Rh antigens are relevant. Now, what are the safety which is required for choosing a donor for blood transfusion? First is the recipient's plasma should not contain any antibody that will damage the donor's erythrocytes. Second, the donor plasma should not have any antibody that will damage the recipient's red cells. Third, the donor red cells should not have any antigen that is lacking in the recipient. Means, if the transfused cells possess a foreign antigen, it will stimulate an immune response in the recipient. Okay? So that will cause some adverse effects. Now, ideally speaking, the donor and recipient should belong to the same ABO group. It is used to be held that the O group cells should be transfused to the recipients of any group as they possess neither the A nor the B antigen. So, O group were designated as universal donor. Now, the anti-A and anti-B antibodies in the transfused O blood group do not ordinarily cause any damage to the red cells of A or B group recipients because they will render ineffective by dilutions in the recipient's plasma. Now, if some O group plasma may contain isoantibodies in high titers like 1:200 or more than that, then they will damage the recipient cells. Okay, and they are known as the dangerous O group. Fine. Now, due to absence of isoantibodies in plasma, the AB group persons were designated as universal recipients. You all know that. Fine. So, AB group donors may not always be available due to their rarity, and it may on occasion be necessary to use donors of other groups. Fine. Rh compatibility is important only when the recipient is Rh negative. Now, how? An Rh positive person may safely receive either Rh positive or negative blood, but an Rh negative individual receiving Rh positive blood may form antibodies against the Rh antigen. Now, subsequently, it will result in what? Adverse reaction. Okay. Now, an additional risk is to the woman who is Rh sensitized, and then she is leading to hemolytic disease of the newborn. Therefore, it is particularly important that Rh negative women who are not past childbearing age receive only Rh negative blood. ABO grouping and Rh typing of the donor and recipient, it is invariably necessary before transfusion to perform a cross matching to ensure that the donor's blood is compatible with the recipient's blood. Fine. Now moving ahead. Next, hemolytic disease of newborn. Now what happens? When an Rh negative woman carries an Rh positive fetus, she may be sensitized against the Rh antigen by the passage of fetal red cells into maternal circulation. Now, minor transplacental leaks may occur anytime during pregnancy. Okay. But during delivery, the fetal cells enter maternal circulation in the large number. During a subsequent pregnancy, Rh antibodies of the IgG class pass from the mother to the fetus and damage its erythrocytes. So, it's causing an adverse effect. Hemolytic disease does not affect all the offsprings of Rh incompatible unions. Its incidence is much less than expected. Okay. Now there are some factors which influence the incidence of hemolytic disease due to Rh incompatibility. Now how? Immunological unresponsiveness to Rh antigen is one reason. Second is feto-maternal abio incompatibility. Third is number of pregnancies. And fourth is zygosity of the father. Immunological unresponsiveness, what happens? Not every Rh negative individual forms Rh antibodies following antigenic stimulations. Some fail to do so. So, even after that, injections of Rh positive cells are given. So, they are called as non-responders. Fine. Now, feto-maternal abio incompatibility. What happens? Rh immunization is more likely to result when the mother and fetus possess the same ABO group. When Rh and ABO incompatibility coexist, Rh sensitization in the mother is rare, and so in this situation, the fetal cells entering the maternal circulations are believed to be destroyed rapidly. Okay, understood? And they are destroyed by whom? By ABO antibodies before they can induce Rh antibodies. Third, number of pregnancies. The first child usually escapes disease because sensitization occurs only during its delivery. The risk to the infant increases with each successive pregnancies. Fine. Now, zygosity of the father. An individual may be homozygous or heterozygous with respect to the D antigen. When the father is homozygous, all his children will be Rh positive. When he is heterozygous, half his children will be Rh positive. Fine. Next, detection of Rh antibodies. Most Rh antibodies are of IgG class, and being incomplete antibodies, they do not agglutinate Rh positive cells. Fine. And in minority class, IgM class antibodies are present, and these are not relevant in the pathogenesis of hemolytic disease as they do not transverse the placenta. Fine. So IgM class is not involved in pathogenesis of hemolytic disease. Understood? IgG class anti-D antibodies may be detected by the following techniques. First is using a colloid medium such as 20% bovine serum albumin. Second is using red cells treated with enzymes such as trypsin, pepsin, physin and bromelain. Third is by the indirect Coombs test. Now we will not understand these techniques or the methods. Fine. We are just looking at the applications. Now moving ahead. Next is identification of Rh incompatibility. Rh typing should form part of routine examination. Okay. As discussed earlier also, when the woman is Rh negative and her partner is Rh positive, fetal complications are expected. Fine, now understood. So, women with Rh incompatible pregnancies should be screened for Rh antibodies by the indirect Coombs test. We will not go for the test, will not understand what exactly happens right now. Fine. Now, when hemolytic disease is diagnosed, antepartum intrauterine transfusion with Rh negative blood may be indicated. So, now that is understood. Fine. Red cells introduced into the fetal peritoneal cavity will find their way into circulation and will survive normally. Okay. When a baby is born with hemolytic disease, exchange transfusion with Rh negative ABO compatible blood is the treatment of choice. So it depends. Next, prevention of Rh isoimmunization. Now what happens? What is isoimmunization? A condition that happens when a pregnant woman's blood protein is incompatible with the baby's, causing her immune system to react and destroy the baby's blood cells. Fine. Now, remarkable success has been achieved in the prevention of Rh isoimmunization by the administration of anti-Rh IgG antibody at the time when antigenic stimulation is expected to take place. The recommended practice to inject 100 to 300 microgram of Rh immune IgG to an Rh negative woman immediately after delivery is preferred. Fine. Next, ABO hemolytic disease. Now what happens? Maternofetal ABO incompatibility is very common. Okay. Now in persons of blood group A or B, natural antibodies are IgM in nature and so do not cross the placenta to harm the fetus. Fine. However, in persons of blood group O, the isoantibodies are predominantly IgG in nature, hence ABO hemolytic disease is seen largely in O group mothers bearing A or B group fetuses. Understood? Now, ABO hemolytic disease, as we understood that it is largely seen in the O group mothers. Now, what happens? It may also occur even in the firstborn without prior immunization. Okay, so ABO hemolytic disease is much milder than Rh disease, probably because erythrocytes of the newborn have fewer A or B antigenic sites as compared to the adult erythrocytes. Now for this, direct Coombs test is performed. Will not go for the test. Moving ahead. Blood group and diseases. Now what happens? It has been shown that some diseases may influence blood group antigens. Blood group antigens have been reported to become weak in leukemia. It's an example. Okay. Next is several studies recently described the involvement of ABO blood group in the pathogenesis of many human disorders, including cardiovascular disease and cancer, so that its clinical significance extends now beyond the traditional boundaries of transfusion medicines. Now let's understand how, as discussed, human disorders including heart attack and heart disease, Then second is brain function and memory loss. And third is cancer. Okay. So now we'll understand how they are involved in this human disorders. Fine. Now ABO gene. We have studied the inheritance of the ABH antigens. ABO genes, if they are present, okay, if ABO gene is present, they are involved in our immune system's functionings, okay, causing, some of them causing like heart attacks, some of them causing some other diseases, some causing brain function loss, some causing memory loss, some are causing different types of cancers. Now for example, a red cell suspension, which is contaminated with certain bacteria such as Pseudomonas aeruginosa, becomes agglutinable by all blood group sera and even by normal human sera. Now what is this phenomena? It is Thomson-Friedrich phenomena. But what it does is it agglutinates normally and then causes a person suffering with some systematic bacterial infections. Okay. Now what it is called is T antigen, anti-T agglutinants. Okay, they are normally present in human sera. Fine. Now several investigators have attempted to correlate blood group and susceptibility to certain diseases. Fine. Next, forensic aspects. Forensic applications of blood group studies is of great value in detection of crime, as we know, and determination of paternity is another spine in forensic medicines after studying the blood group serology. Understood? We are not going in much detail. These are the applications where the ABO groups are involved. Fine. I'll upload all the study material on the Google Classroom. Please go through it. If you have any doubts, please ask. Okay, start reading. Thank you.